G'day, welcome to DNA Lives Forever, where I talk about various topics related to DNA, health, and/or biological immortality. I'm David A. Elliott, and this is the second of 3 episodes where I talk about genetic research into the deaths of Kathleen Folbigg's 4 children, for which she was sentenced in 2003 to a minimum of 25 years in prison and labelled as Australia's worst female serial killer. Recent genetic research indicates that at least 2 of the children most likely died from natural causes due to rare genetic mutations which resulted in her pardon and release from prison this year on the 5th of June 2023. In this second episode, I will focus on discussing the most recent research and how it may provide a natural explanation for the deaths of Sarah and Laura. But before I do, I will summarise the main points covered in the first episode. Kathleen Folbigg had 4 children who died as infants from 1989 to 1999. In isolation, all the deaths appeared to be from natural causes, with no physical evidence to indicate foul play. Investigations into possible genetic causes began after the death of her second child, Patrick, in 1991. During the 1990s and beyond, to test for a genetic cause required identifying any known genes that may be involved and then testing for any known mutations in that gene. This is very limiting as actual genes or mutations involved may be unknown or not identified. So not finding anything would still leave open the possibility of a genetic cause. No genetic cause was found at the time, and after the 4th death, police began to investigate, and based on circumstantial evidence, she was charged, convicted, and sentenced for killing all 4 of her children. At this point, it may be appropriate to elaborate on a statement I made in the first episode where I said, "It seems the last doctor was reluctant to make a determination of the cause of death due to the other 3 deaths and referred the matter to police." The doctor was called Dr. Carla and was the forensic pathologist who conducted the autopsy on Laura. In a letter to Singleton Police dated 29 June 1999, 4 months after Laura's death, he appears to strongly argue that natural causes of death for all 4 children are unlikely and homicide should be investigated. A particularly relevant part of the letter states, "I do not believe that SIDS runs in families, but other conditions may do so. Many natural diseases may cause death during infancy, and these seem to have been excluded, either during life or at post-mortem. The deaths of all 4 children in the one family in Australia in 1999 is not only extremely puzzling, but clearly warrants thorough investigation. In my opinion, the possibility of multiple homicides in this family has not been reasonably entertained, investigated, and excluded. If acts of foul play have been committed, it is most likely that these acts have been in the form of deliberate smothering." He then notes that there are no specific post-mortem findings of this, but smothering may leave no trace. In his concluding comments, he states, "In my opinion, the deaths of the Folbigg children should be investigated as a single unit. I am greatly concerned by the deaths of these children, and I believe very vigorous efforts of inquiry should be made to exclude homicide in any or all of these children." Investigating all 4 deaths as a single unit also appears to be the approach he took in his final autopsy report for Laura dated 13 December 1999, where he drew similar conclusions of homicide by deliberate smothering and excluded Laura's myocarditis as a less likely cause of death. Also of note in Dr. Culler's letter to Singleton Police is where he states, "There remains an exceedingly remote possibility, and so remote to be almost impossible, of some underlying and undiagnosed genetic or metabolic disease which has so far not been detected." Well, moving forward 20 years, it seems an underlying and previously undiagnosed genetic disease has now been detected. But before discussing this, I will add that in a letter dated 27 February 1992, the Folbigg's clinical geneticist, Dr. Colley, indicated that even though a diagnosis was not made, that the risk for a similar outcome with any future children they had could be as high as 25%. I assume this was based on the possibility that the same recessive gene carried by each unaffected parent leads to a 1 in 4 chance of having a child with an undiagnosed genetic condition. Now, just as a personal aside, about 20 years ago when I did my Masters in Genetic Counselling, so around 2003, to make such a assumption about it being up to 1 in 4 would have been standard if someone came in and they were looking for a possible genetic cause and nothing was found. It'd be basically, well, it's could be more than population risk because you've already had a child that's been affected, and up as high as generally the recessive condition for which you've just talked about. But anyway, I'll continue. Since Kathleen Folbigg's conviction in 2003, gene technology has advanced in leaps and bounds. This includes the area of gene sequencing and has meant that in recent years it has become possible to sequence all of a person's DNA, known as whole genome sequencing, and then work in reverse when searching for a genetic cause. That is, once all of a person's DNA is sequenced, it is then possible to identify all the mutations, identify the genes that contain those mutations, determine if the function of those genes is known and the effects of mutations in those genes, and if any of these could explain the condition or death being investigated. Now, before I go on to discuss the testing and analysis in relation to Kathleen Folbigg and her children, it should be noted that one of the investigators, Professor Vinuesa, has stated that she considered, prior to carrying out this genetic investigation, that even if there was a genetic cause, the probability of finding it would be less than 50%. I expect part of the reason for this is that the function of more than half of all genes is still unknown, but also it could be due to the quality of the children's samples available for testing. The genetic investigation began in 2019. Kathleen Folbigg willingly provided a sample, but her ex-husband refused. It was thought not having his DNA may reduce the ability to find a possible genetic link, but ultimately it appears this was not the case. For the children, testing was conducted using either their stored newborn screening heel prick sample card or autopsy samples. Testing for the last child, Laura, was restricted to a less complete technique called whole exome sequencing due to microbial contamination of the sample. While there may be many mutations identified throughout our DNA, most will have little, if any, effect on us. So, once the DNA sequencing was completed and the mutations identified, a process to decide which ones were irrelevant and unlikely to be related to the children's deaths was begun. This left a number of genes for further investigation to consider the likelihood that the mutations found could be related to the children's deaths. It was found Kathleen Folbigg and her two daughters, Sarah and Laura, had one mutation in a gene called CARM2 that could reasonably be considered to have caused the two girls' deaths. At the 2019 inquiry, there was disagreement by some expert witnesses about this mutation being responsible, but further research and evidence had reinforced that there is a reasonable likelihood it caused their deaths. Now, I'll try to explain this without getting too technical. To start with, it is not unusual for there to be multiple copies of the same gene in our DNA. CARM2 is one of 3 copies of the CARM gene, the others logically being called CARM1 and CARM3. In simple terms, these genes are involved in the regulation of heart contractions. The first mutation in one of these genes was identified in a Swedish family in 2012 and associated with cardiac arrest in infants in 2013. Up to 2020, only 74 people had been identified with mutations in these 3 genes, but there were 35 different mutations. A number of these mutations have been associated with cardiac arrest in children and infants, including whilst sleeping. The mutation in the Folbigg family was a new one for this gene. However, the published research paper on this investigation noted that recently 2 young children in a family with an identical mutation in the CARM3 gene had cardiac arrests, one of whom died. Further research confirms that Folbigg's CARM2 mutation would be no different to this family's CARM3 mutation. The question then arises, if this is the case, why isn't Kathleen Folbigg affected by this gene? The reality is that the effects of a gene can be influenced by other genes, called modifier genes. It was thought possible that Sarah and Laura may have inherited modifier genes from their father that made the effects of this gene mutation much worse. Such a gene has recently been identified in them, called REM2G96A or REM2G69A, and has been confirmed to aggravate the negative effects of their CARM2 mutation. This modifier gene was present in all 4 children, but not Kathleen Folbigg. It was also considered possible that Kathleen Folbigg may have had modifier genes that reduced the mutated CARM2 gene's effect, but not pass these on to Sarah or Laura. Similar situations have been observed with other families with the CARM gene mutations, where some members of the family with the gene mutation appear unaffected, but cardiac arrests and deaths occurring in others as young children. In any case, Kathleen Folbigg does have a medical history of fainting episodes that have taken more than 30 minutes to recover from, including observed instances whilst in prison, and it is thought they may be a result of having this gene. Let's now focus back on Sarah and Laura. They both had respiratory infections leading up to their deaths, and the recent researchers considered such an infection in association with this gene mutation could trigger serious cardiac arrhythmias, which is an abnormal heartbeat, and resulted in their deaths. Sarah and Laura were the 3rd and 4th of the children to die. Sarah died in 1993 at 10 months. She also suffered from sleep apnea, so her breathing would sometimes stop and start during sleep. And her autopsy indicated high levels of bacterial infection in her lungs, with her death originally recorded as sudden infant death syndrome, SIDS. Laura died in 1999 at 18 months. Kathleen Folbigg found Laura when she was blue with a slow heartbeat. Ambulance reported they detected an agonal heartbeat, meaning it was very slow and irregular, but were unable to revive her. Her autopsy indicated high levels of bacterial infection in her spleen, myocarditis, inflammation of the heart muscle, and petechial hemorrhages in the thymus. Now, these are small red blood spots on the thymus, and it is observed in 87% of SIDS cases. At the time, her death was recorded as undetermined and not as SIDS, as she was more than 12 months old. The recent research notes that myocarditis is considered a major cause of sudden cardiac death in those under 20 years old. It was also noted that Laura was treated with pseudoephedrine phenylephrine, which is known to trigger cardiac arrhythmias, and a combination of all these factors can explain her death. Now, this could explain the deaths for the 2 girls, but what about the first 2 deaths for the boys? There could be another gene involved here that the researchers did not include in their original analysis. However, they do mention it in supplementary material to their published research paper. But I will leave further discussion of this for the next and final episode. And this brings us to the end of this episode of DNA Lives Forever. I hope you enjoyed learning about how analysing the results of whole genome sequencing led to identifying the CARM2 gene in Sarah and Laura, and along with other factors, made natural causes of death a reasonable likelihood. If you like this episode, make sure you follow DNA Lives Forever to listen to the next episode on this topic and hear me talk about other interesting topics related to DNA, health, and/or biological immortality.